So we had just finished discussing the differences between arteries and veins. And the intervening structures are capillaries. You know the order through which blood flows within the vasculature. Capillaries are in the middle. Their job is to exchange nutrients, substances. And they're very, very small. They are 1 cell width thick, 1 layer of endothelium with a basement membrane, and that's it. So very small, very thin, microscopic. You can't see them with the naked eye. And I showed you last class that these were about the size of a red blood cell. In fact, a red blood cell has to squish itself in a little bit. They're a bit bigger than the capillaries. And they can only fit in single file, one at a time. So single layer of cells, basement membrane, and it's because they're so thin that they do the job of exchanging substances so well. Arteries and veins with 3 layers. This only has the tunica intima. With 3 layers, and thick layers don't do the job of exchanging substances very well at all. That's the role of the capillaries. And while we look at one capillary tube here, and you can see the one, the lumen, the opening through which the red blood cells would travel, and the singular wall on the outside, they are not isolated. In fact, they often group together and form a meshwork around and into tissues in the body. In this figure, it looks two-dimensional, and it's to get the point across that arteries merge with veins through a capillary bed, a network of capillaries. There's probably millions of capillaries within the entire capillary bed system in the body. They're found near almost every cell in the body because every cell needs to exchange substances. There are a few notable exceptions that we talked about last year, like the cornea and the lining epithelium. Those don't have specific capillary beds, but typically most cells, the rest of the cells, do have a capillary bed in close proximity. And that's because their job is to make the largest surface area possible. The largest surface area possible in very close contact with all cells of the body. Largest surface area possible. Why? Why the largest surface area possible? It's simply because that gives it greater opportunity to exchange substances. More surface area, there are more places where substances can cross. Which is good because these substances, as we'll see in a video coming up, are really only crossing by simple diffusion. Remember, so much in physiology is gradients. Things move across capillaries according to concentration gradients, for the most part. So largest surface area possible in close contact with cells gives numerous locations for the exchange of substances. Now, these are conveniently organ-shaped, these capillary beds. You can see the heart that we know well and we've studied at length. You can imagine these are probably the lungs. They're in close proximity and they look vaguely lung-shaped. We have the liver when the slide comes back on in the bottom left. If it comes back on. There we go. Does that happen a lot? Because I don't get to see it. I'm always facing you. Is it for long periods of time, or is it just generally quick flashes? Okay. Second or two. It's enough to make you notice, to pay attention, maybe, to be disrupted. I don't know how I can fix that, but we'll leave it for now. Maybe it's to change some of the hardware. But anyways, the liver, stomach, spleen, they're conveniently body-shaped, And that's not to say that the capillary beds themselves are organ-shaped. I just said body-shaped, but I meant organ-shaped. They're not organ-shaped. They perfuse and wrap around and twist and turn around all the cells within those organs. This is just to convey the idea of the capillary bed. Now I want to make two more points that aren't included here, and then we'll move on to the video. Point one is that the size of the capillary bed changes with the demand, with the metabolic demand. The size of the capillary bed changes with the metabolic demand. So if the tissue in question has a high metabolic demand, maybe the brain or the muscle can have a high metabolic demand, those capillary beds will be large. They will be large. If a tissue does not have a high demand, a high metabolic requirement, the capillary bed is often quite small. And so it's proportional to the tissue. Second point, the capillary bed can open up. It can open up with demand. So muscle might have a really large capillary bed, but as you sit here at rest, you're not engaging your muscles a lot. You don't need to deliver blood to the complete capillary bed. Some areas are shut off. Blood doesn't go into all the nooks and crannies. But as you get up, walk around, move to your next class, go for a run, and the muscle becomes more active, more fibers require blood, the capillary bed opens, and blood goes through the entirety of the capillary bed. So size is proportional to the metabolic demand, and the capillary bed can open up with that demand. Now, being microscopic, it's often hard to imagine blood flow through a capillary bed and the transfer of substances. Actually, first what I want to do is show you what it might look like. Just in theory, we know this is a continuous system. Arterial blood becomes venous blood in the capillary bed, and you can see this is an example of a node or a very small bed with arterial arteries on the left, arterial blood flowing on the left, and as it enters the capillary bed, you can see the gradient change from oxygenated red to deoxygenated blue as substances are exchanged. And notice it's not only in two dimensions, wrapping around all theoretical invisible cells in this example, as the blood then collects and pools inside the veins and pumps back towards the heart through these one-way valves. So nice to be able to see the 3D model, and the layers you can imagine. I'm not going to go through them in depth. The vasa vasorum you can see. It's a nice way to put it into context, I think. That 2D model I hope the volume works. A 2D model doesn't show the movement of substances or the movement of red blood cells. So that's what we're going to look at in this 3D physiology video. You have the link in the slides on Moodle as well, but we're only watching the first section.
The survival of every cell in our body depends on its ability to obtain the gases, nutrients, and other substances needed to complete all of its chemical processes. Cells also produce byproducts and wastes that must be transported away from the cell. The transfer of substances between the interstitial fluids surrounding each cell and the blood flowing past the cells in the capillaries is referred to as capillary exchange. Nutrient and oxygen-rich blood reaches cells by passing through arteries which branch into increasingly smaller vessels, eventually becoming arterioles that divide to become capillaries. The capillaries recombine into venules and then to veins as they return low nutrient but higher in carbon dioxide blood to the heart. Capillary walls are composed of thin squamous cells, which in most cases have pores through which some substances and fluid can pass. This allows the exchange of substances between the bloodstream and the interstitial fluid. Simple diffusion is the primary method for exchange of many substances through the capillary wall. Diffusion from the bloodstream into the interstitial fluid, or vice versa, is solely dependent upon the substance's concentration gradient on either side of the vessel wall. Lipid-soluble substances and gases can pass directly through the thin cells of the capillary wall. Lipid-insoluble substances diffuse through pores in the capillaries. Changes in diffusion gradients of nutrients, gases, and wastes may be the result of aerobic cellular respiration within each cell. Active cells consume oxygen during the production of ATP in the mitochondria. As more and more of the intercellular oxygen is used up, its concentration becomes less than that of the surrounding interstitial fluid, causing oxygen to diffuse into the cell. As the oxygen level in the interstitial fluid decreases below the concentration in the bloodstream, oxygen diffuses out of the capillaries into the interstitial fluid. Carbon dioxide is given off during ATP production. When the accumulation of carbon dioxide within a cell exceeds that of the interstitial fluid, the carbon dioxide diffuses out of the cell. As the carbon dioxide level in the interstitial fluid increases to a higher concentration than in the bloodstream, the carbon dioxide diffuses into the bloodstream. Transcytosis provides a method to move larger molecules, such as proteins or...
We're not going to learn about that. I really like, though, that it's a deliberate, stepwise description of diffusion. And it makes a lot of sense. You're probably sitting there thinking, of course. But I think it really does highlight and describe the flow of substances properly. Those substances, in this case, being oxygen and CO2. Nice to connect back to the first semester as well, when we talked about metabolism ever so briefly. You will learn much more about that as we move to XPhys in your second year. So diffusion gradients with the concentration gradient is how capillaries exchange substances. And the same thing would go for glucose, cholesterol, free fatty acids, anything else that needs to be delivered to the tissue. So with the major components under our belt, you've seen something like this already. The distribution of blood when at rest is heavily weighted towards the veins and the venules. 2/3 of the blood at any one given point in time are stored in the veins. And I suppose it's because they needed a job. The arteries are pressure reservoirs. The capillaries are exchange conduits or exchange vessels. And the veins and venules are blood reservoirs. So 2/3 of the blood at any one time is stored there. And it's really because the veins are very compliant and supple. They can expand to increase volume and accommodate more blood. But this isn't static. This can change. In times when we need blood for whatever reason, we can mobilize, quote unquote, blood from that compartment to others. In cases like hemorrhage, if you're losing blood quickly, or during exercise where you need more blood to be supplied to various tissues, we can mobilize blood through a process called venoconstriction. So you've heard of vasoconstriction. It's the same thing, but applied to veins. Vasoconstriction applies to arteries, venoconstriction applies to veins. We often use those interchangeably. So we have some mild ability to contract the veins, to squeeze the venous side of the vasculature, and reduce the blood that's stored here. And all that does is it pushes blood into these other compartments as needed. Let's leave it at that for now. So distribution of blood, and I want to bring it all together as we move into the physiology of blood flow through the veins in coming weeks. With this slide, to appreciate not only the difference in structure between the different components, but how that structure impacts function in those different components. So what we're looking at here are a dual trace of both speed of blood flow, or velocity of blood flow, and the cross-sectional area. So cross-sectional area is just if you took all of the vessels of a certain type and stacked them up in front of you and cut them, all of the combined area of those vessels. So the aorta, very small cross-sectional area. It's the largest artery in the body, but there's only one. Many more arteries, many more arterioles, and almost infinitely more capillaries make for a very large cross-sectional area there. So, arteries we know are pressure reservoirs, and you can see that pressure, at least as it translates into velocity. There's a fast transmission of blood in the arteries. They don't have a large cross-sectional area because they're conduits. They get blood where it needs to go. And they do it relatively quickly. And that pulsatile wave nature of that velocity graph simply represents every heartbeat. With every ejection, blood speeds up and it slows down between beats. And it speeds up and it slows down between beats. But overall, there's a general progression where we have fast blood flow initially that tapers off. Arteries are conduits for blood to get it where it needs to go. Now it needs to go to the capillaries to exchange nutrients, as we just saw. At the capillaries, we've already mentioned that there is a large cross-sectional area, making the largest surface area possible. And just appreciate how many-fold larger this is than either veins or arteries on either side. Orders of magnitude larger. And because there's so much space for the blood to spread out, pressure drops and speed or velocity drop. Now, why is it important to have a low velocity moving through the capillaries? It's important to give time for things to exchange. Time to offload oxygen, time to onload CO2, time to deliver glucose, time to deliver hormones, time to pick up hormones. Really low velocity at the capillaries because of their large cross-sectional areas, or area, period. And then in the veins, as the venules merge and anastomose together, they gradually converge more and more until we end up with only 2 veins entering the heart, maybe 3 if you were paying attention last class and in lab. We have relatively low cross-sectional area again and some velocity as we push blood back to the heart. So structure dictates function largely in this graph, and we'll come back to this later. So as we round out this section, I wanted to go all the way back. Remember in the intro I laid out a few learning objectives, and this is that slide again. I just want to see if we've done a good job of learning about the components of the cardiovascular system. I think yes, we know the surface features of the heart, we know the internal features of the heart, we know some of the major vessels, at least we know the types of major vessels, if not their names and their destinations yet. That's coming up. We focused on the anatomy of the heart. We didn't focus on physiology, so I crossed that out. That is coming up. You knew before you came in, but we've mentioned that the heart pumps blood, which is a function, a physiological function of the heart, but we don't know how yet. That exploration is coming probably next week. Contrast the structure and function of arteries and veins. Absolutely, we did some of that today. And then this whole fourth point, I'm moving to the next section. We didn't look at major circulatory paths, but we will get into that shortly. So we're just dotting the I's and crossing the T's. What I would say is that in place of number 4, I've added this fifth one. We've described a general route through the typical types of vasculature in the body, not a specific route. I'm trying to hold myself accountable. So let's summarize then, which is sort of what we did. But we've determined the heart. We've determined the external surface features of the heart that allow us to determine its orientation. You know the major vessels. You know some of the major components, the external features of the chambers of the heart. We know the heart doesn't sit alone within the thorax or in the mediastinum. It's enveloped in a double-walled sac that's job is to reduce friction during normal wear and tear, allowing normal contraction to proceed with minimal obstruction or opposition. With the surface features down, we moved inside, and we know the 4 main chambers. We know the ordered flow through those chambers. And we know the valves that divide or regulate entry into those chambers at very important junctions within the heart proper. Not only do we know the names of the valves, but we know how many cusps they have, how they open and close. We know what auxiliary structures there are, or accessory structures help them open and close and be stabilized. We know their job. Valves are there to regulate flow, specifically to ensure unidirectional flow. Make sure there's no backflow through the heart. And all of that is due to the generation or creation of pressure gradients across that valve. Pressure dictates if the valve is open and closed, open or closed. It dictates if there is movement of blood between compartments, how quickly it moves between compartments, and as we get into the next section, where it goes in the body. Now, all of these points are not really satisfying. They're all purposefully vague because to get into detail on any one point would mean reviewing slides or pages of the lab manual, which is your responsibility in class, in lab, and outside of class, learning these details. But we can do this fairly effectively from the material that we've covered here.